All right, good afternoon. Thank you so much, particularly Dr. Chen Chunyu for the invitation. Today I will show you some work which was published not a long time ago about the endothelial UCP2, this uncoupling protein 2 is a new mechanosensitive suppressor of atherogenesis. Now we have been interested in the impact of flow pattern, the shear stress due to the blood flow and on the endothelial function. And basically, apart from hydrostatic pressure, and actually it's blood flow generate two types of shear forces on the endothelial cells. Now one is we call laminar flow, mainly impacting the endothelial cell on the more straight part of blood vessel endothelial cells, and which normally considered to be beneficial. And another one we call the oscillatory shear stress or disturbed flow, and occurring in the vascular curvature or branching points of blood vessel. And this is actually well demonstrated. Now, in this curved blood vessel normally has higher level of inflammation, and then also the deposit of the atherosclerotic plaques are more often identified in this area. But for so many years, people try to understand now how this the mechanostimulation affect the phenotype of endothelial cells, and basically how this turbulent flow now trigger the inflammatory response in endothelial cells. So since about 2014, I think that time, about 10 years ago, and then we started spending more time on this area, and we try to understand all those inflammatory the markers or these proteins which are closely associated with now the inflammatory response in human endothelial cells. And today, I want to share with you this one study by former the PhD student, and later on spent a couple of years in the lab to complete the study about UCP2. Actually, back to about 2012, and our first publication, and they show very clearly if you have global overexpression of UCP2. Now again, UCP2 actually is a proton carrier expressed by mitochondria along these proton gradients. And this is very important to control the production of reactive oxygen species now along this electron transport chain. So a long time ago, we found in the diabetic mice, if you overexpress UCP2, and very effectively increase endothelial function, mainly by increasing increasing the enos activity, and meanwhile suppressing the production of ROS, or suppress the oxidative stress. But today, I think this is another interesting because we've been studying now how this flow pattern affects some important intracellular regulators, particularly in the production of ROS and the inflammation. So we have this setup called ABIDI. We can culture human endothelial cell in our setup, and then we can mimic, there's two types of blood flow or shear forces. One we call the disturbed flow, which is supposed to be detrimental, is harmful to our endothelial function. Another is the laminar flow, which normally produce the beneficial effect. Now, USS, that means unidirectional laminar flow. That's good. And then these cells exposed to the USS or laminar flow, you can see the obvious the upregulation of UCP2 expression in endothelial cells. And by the contrast, if you expose cells to the oscillatory, the disturbed flow, which produce bad effect, and then you can see there's a significant reduction in the UCP2, the expression. So that gives a very clear indication in our early studies. And also we have an emphasis, standing data able to show actually there's a different level of UCP2 expression in the straight blood vessels or in the curved blood vessel. in terms of the endothelial cell. Now, this is what we did. Now, normally, now this straight part, laminar flow is good. And then we can stand these mitochondrial produced by the mitochondrial RS, use these mitosocs. You can see very clearly the aortic arch, which actually experience disturbed flow, now has a higher level of this RS as compared to the endothelial cell now located in the more straight, the thoracic aorta. And then also, if you use a Western blot, get this part of these arches, and you can see very clearly. Now, this aortic arch in this area, you can see now increased expression of VCAM1, which actually is one of those initial factors leading to the development of atherosclerosis. Now, this is correspond to the reduction in the UCP2 expression, and the same as a similar pattern change in the messenger RNA expression for UCP2. And we of course, because the UCP2 is not transcriptional factor, and then we try to understand how this UCP2 expression can be induced now by exposing our cells to laminar flow. And then we're screening a number of possible now mechanosensitive transcriptional factor, and then we pick up this KLF2. This is well known, yeah, and this is known for many, many years. And then we happen to know now this the KLF2 actually is upstream regulator of UCP2, and this is something new from our study. So you can very briefly you can see now. If you have the disturbed flow, if you overexpress UCP2, you can see now suppress now UCP2 expression due to disturbed flow can be fully reversed. Or if you have the laminar flow induced upregulation of UCP2 can be suppressed if you suppress KL2 in the endothelial cells. That's very clear. And of course, we study a bit more, and then we try to understand how these things work. And here, very clearly, due to the time constraint, just a little bit information for you. Again, we manipulate human endothelial cells. Now, in this case, we overexpress KF2. For example, now you can see marked increased expression of UCP2, and the same as the eNOS. And the KF2 is known to increase now the transcription of eNOS, and that's important to the validate. Now this is important and linked to the endothelial function. And of course, because this you design the drugs, and normally I would like to test those drugs we have been using for many, many years, and then we choose a few. And then one, of course, is our favorite drugs. It's a statin. We use rosuvastatin and the semivastatin. Another is the resveratrol. And then you can see these are both we showed before already able to increase the KF2. And then here, if you expose your cells or treat your human endothelial cells with either of those drugs, you can see very clearly. And then they are capable of increasing expression of UCP2 messenger RNA, also the protein level. But by contrast, if you treat now human endothelial cell with this inflammatory cytokines such as interleukin-1 beta, and it's quite effective to suppress its, the expression. So this is further can be confirmed. We can, if you note down the KF2, and then all this effect produced by resveratrol or statins on the expression of UCP2, basically reverse, reverse. And then of course we need to, in the animal studies, and then in order to the accelerate formation of plaques, we also use upper knockout mice, and then in this case we knock down the endothelial cell UCP2 and inject it with the PCSK9 overexpressing virus, and then just try to speed up, this fasten the formation of plaques. Now, for some time, you can see very clearly, of course, we measure those lipids, all those things, and see, and if you specific note down of endothelial UCP2 actually result in increase now the plaque formation. So this can be confirmed because, as I mentioned, UCP2 now in our study, because this is another new mechanosensitive driver or the mediator now of this vascular function. Of course, we use another model called this partial ligation of carotid artery, and this only for the short duration of time, normally 3 weeks. We can kill the animal. And then what happened? We ligate here partially, and then what happened? Your blood flow not so smooth, and then they will generate this turbulent flow, just equivalent to the oscillatory shear stress. Now this is why this the spot, and that actually is a deposit of plaques. And this you can see very clearly, even in the carotid artery, if you specific, the suppression of UCP2 result in increase the plaque formation. Or on the other hand, if you use the EC specific overexpression of UCP2, and actually can reduce now the plaque formation in these models. And of course, you want to ask now, how these things take place? And we have this try to knock down those endothelial cells, and then subjected to the single cell sequencing analysis, and we found there's a few pathways as pop up. One is FOXO signaling pathway, another is the AKT signaling pathway. These both pathways actually is related. And you can see very clearly now this is if you knock down the UCP2, and then you will see this as increase now FOX1. And now this is FOX1 either in the cytosol or in the nucleus. Now to save the time, and then we spend some time to try to understand whether or not if this is the case, now if you suppress UCP2 on endothelial cell leading to activation of FOX1, and then FOX1 inhibitor should be able to do something. So we treat this animal model with this FOX1 inhibitor, and indeed you're able to see now there's some beneficial effect to suppress now there's a plaque formation due to the UCP2 always the knockdown in endothelial cells. Another thing is that we know if you knock down the UCP2, then what will happen? Because the mitochondria might not be able to produce enough ADP, at the same time ADP might increase. So the ratio might go to turn on. Now that's important, the kinase called MPK. So you can see very clearly this is MPK, but compound C actually is MPK kinase inhibitor. So you can see if you use the UCP2 overexpression, and then you will see this is increased phosphorylation of FOXO1. That means will be degraded, will suppress, and very little will able to be translocated into nucleus. And also this negative regulator going to be upregulated. So we did quite a lot of work, and then figure out how these things might work. In the end, probably in this signaling pathway, UCP2, and then somehow if you the suppression in endothelial cell and able to increase MPK, overexpression, sorry, the MPK, and then suppress now FOXO1. But FOXO1 associated with inflammation is known. Yeah. So just in seconds, to make a small summary here. And I think we provide very clear evidence, show this endothelial cell UCP2 actually is a new mechanosensitive regulator of inflammation, even that situated in the mitochondria. That certainly there's some other, the mechanisms still unknown to us, but we have these few drugs which are being used, yeah, either for treating patients with hyperlipidemia or coronary artery disease, or acting as a health supplement such as the resveratrol, and actually able to increase the UCP2 expression, now mainly through activation of KF2. So KF2 certainly is very, very important. We have a couple studies in this area. So this actually is my former student, now back to Zhejiang University, working in one of hospitals, and that's actually one of his thesis work. Thank you so much.